Welcome everyone. In this chapter, we are going to study protein. Protein are one of the most important biomolecule in the human body. They help in body structure, growth, repair, and many life function. Let us start step by step. Before starting, let us see what we will cover in this chapter. First, we will learn the definition of protein and how protein are classified based on composition and solubility. With example, next we will learn amino acid and their classification based on chemical nature and nutritional requirement. After that, we will study the four level of protein structure. Finally, we will learn qualitative test biological role of protein, and this is related to protein malnutrient. Protein are natural occurring polymer made up to amino acid. Almost everything that happens inside cell involve one of the more protein. Protein provide structure to cell, help in chemical reaction and carry out many important tasks. About 20 amino acids are commonly found in protein and they are called standard amino acids. These amino acids contain an amino group and a carboxyl group attached to the alpha carbon. Proteins are classified in the various ways because proteins are very diverse. Some proteins are perceptual like collagen and keratin. Some proteins are do work like enzyme hormones and transport proteins. So classification helping and understand different protein type of and remember the easily for exam. This slide explains the main classification clearly. Simple protein give only amino acid when they are heterologous. Simple protein can be fibrous, which are strong and insoluble, like collagen in connective tissue and keratin in hair and nails. Simple protein can also globular, which are compact and soluble, like albumin and globulin. Conjugated protein content in a protein part plus a non-protein part called a protein group, such as nucleoprotein, lipoprotein, glycoprotein, and phosphoprotein. Derived protein are formed when protein get particularly broken down, producing proteases, peptones, and peptides. Now we focus on classification of protein based on composition. Simple protein contain only amino acid. Conjugated protein contain amino acid plus an additional non-protein part, which give vessel function. Derived protein are formed due to partial heterologous or modification of protein. This classification is important because many functional protein in the body are conjugated protein. Now we compare globular and fibrous protein. Globular protein are spiral or compact and usually soluble in water. They perform dynamic function like enzyme, hemoglobin, insulin, and antibody. Fibrous protein are elongated and usually insoluble in water. They mainly provide strength and structure like collagen, keratin, elastin, and actin and myosin. Now we learn the globular protein group more clearly. Globular protein include albumin and globulin. Albumin are commonly found in egg, white blood, milk, and muscles. Globulin are insoluble in distilled water but soluble in dilute salt solution. These examples are important because they are commonly asked in exam. The slide mainly supports the previous slide with example and visuals. Remember one simple point: globular proteins are usually soluble and mainly two functional groups, while fibrous proteins are usually insoluble and mainly provide structure and strength in tissue. Now we classify protein based on biological function. Enzymes are proteins that feed up reaction. Many hormones like insulin and growth hormone are protein. Storage protein include casein in milk, ovalbumin in egg white, and ferritin or iron storage. Transfer protein include hemoglobin, myoglobin, serum albumin, and ceruloplasmin. These show proteins are essential for many body functions. Now we move to amino acids. Amino acids are building block of protein. Many amino acids exist in nature, but only about 20 amino acids are used for making protein in body. Amino acids join together with peptide bond to form protein. Understanding amino acids is necessary to understand protein. Of amino acid. Yes, amino acid has an amino group, carboxyl group, hydrogen, and an R group. At all normal body pH, amino acid exceed one deuterium, meaning they have positive and negative charge. This is why amino acid and can act like both acid and base. Now we classify amino acid based on chemical nature. Acidic amino acids include aspartic acid and glutamic acid. Basic amino acids include cysteine, lysine, and arginine. Hydrophilic amino acids include asparagine and glutamine. Hydrophobic amino acids include ligand, isoligand, and valine. Sulfur content amino acids include cysteine and methionine. Aromatic amino acids include phenylalanine and tyrosine. Amino acids can also be classified based on the nature of their slide chain, called the R group. Some amino acids are nonpolar and hydrophobic, and some are polar and uncharged. Some are acidic and negative charged, and some are basic and partially charged. The classification helps us understand protein folding and function. For example, hydrophobic amino acids usually stay inside the protein. The polar amino acids prefer the surface. Now we learn polar uncharged amino acids. These amino acids are hydrophilic and can form hydrogen bond. The many content group like OH, CH, or NH2. Explain include serine, tyrosine, cytosine, asparagine, and glutamine. These amino acids are commonly found on the surface of protein and help protein interact with water. Now we learn charged and aromatic amino acid. Basic amino acid is partially charged at physiological pH like lysine, arginine, and cysteine. Acidic amino acids are negative charged at physiological pH like aspartic acid and glutamic acid, aromatic amino acid and content ring structure like phenylalanine, tyrosine, and tryptophan. Aromatic amino acids are important for protein structure and also give paths which result in come protein test. Now we learn non-polar alphabetic amino acids. These amino acids are hydrophobic and usually found inside the protein structure. Explain include glycine and alanine, valine, leucine, and isoleucine. These amino acids help in forming the stable core of globular protein. This slide again highlights the partially charged amino acids. Lysine and arginine carry partially charged strongly. Cytidine is special because it can gain or lose or proton easily. So it is very important in MCM active side. This amino acid help in ionic bonding inside protein. Now we classify amino acid based on nutritional requirements. Essential amino acids cannot be synthesized in the body, but they are required for normal function. So they must be supplied through the diet. If essential amino acids are missing in food, growth and repair will be affected. Now we learn non-essential amino acids. These amino acids can be synthesized in the body, even though they are called non-essential. They are still needed for protein formation and body function. The difference is only that the body can make them when they needed. This slide shows the list of essential and non-essential amino acids. Essential amino acids must come from protein-rich food like milk and egg, pulse, fish, and meat. Non-essential amino acids can be formed inside the body. For example, it is good to remember at least 8 to 10 essential amino acids. Now we start structure of protein. This has 4 level organization. This level for primary, secondary, tertiary, and quaternary structure. Protein structure is very important because protein function depend on its shape. Even small change in structure can be affected by protein activity. Primary structure is the frequency of amino acids in the protein chain. Amino acids are connected by strong peptide bond. This frequency is decided by genetic information in DNA. If the frequency change, protein function can change and it may be cause of disease. Secondary structure is just regular folding of the protein chain due to hydrogen bonding. The main secondary structure are alpha helic and beta pleated sheet. This structure formed because the backbone CO and NH group created hydrogen bond. Secondary structure helping in the protein become stable and organized. Alpha helic. In alpha helic in polypeptide chain, it is like a spring and into a right-handed cover. Hydrogen bond formed between the NH group of one amino acid and the CO group another amino acid in the adjacent strand. The hydrogen bond stabilizes the helic structure. The alpha helic is commonly seen many proteins. Now we land beta pleated sheet in the structure polypeptide chain and stretch out and placed side by side. There are held together by hydrogen bond between different chain. The structure looks like pleat or fold, so it is called pita-pleat sheet. The structure is also very common in protein. Now we learn tertiary structure. The tertiary structure is formed by further folding of the secondary structure into a 3D shape. This folding is stabilized by hydrogen bond, ionic bond, disulfide bond, and a van der Waals force. Tertiary structure is the final shape, a protein, and decided whether it becomes globular and fibrous. If the tertiary structure is disrupted, the protein can lose function. The slide mainly supports a tertiary structure with a diagram. The key idea is that protein fold step by step from a chain to stable 3D structure. Proper folding is necessary for enzyme activity and binding function. Strong folding can lead to loss of function. Now we learn quaternary structure. Some protein contain two or more polypeptide chain called a subunit. The spatial arrangement of this subunit is called quaternary structure. A common example is hemoglobin which forms rapidly only when all subunits come together. Quaternary structure improves stability and function. Slide support quaternary structure with diagram example. The important point is that subunit interact with each other and form a functional protein. If the subunit separated, the protein may not work properly. Now we move to qualitative tests for protein and amino acid. These tests help in identify protein in the sample. Many tests are based on peptide bond or specific amino acid group. These tests are common in practical lab and exam. Now we study the Purette test, which is a common test for protein. In alkaline medium, copper ion react with the peptide bond and they produce a violet or purple color. The purple color indicated the presence of protein. This test is possible for compound that have two or more than peptide bond. The slide explains the test related with the phenolic group of hydrogenic proteins reacted with reactive reagent to give a red color. So a red color indicated the presence of hydrogen in protein. This shows that CAM test detected the specific amino acid group inside the protein. Now we study the biological role of protein and amino acid. Protein provides amino acid during digestion, and these amino acids are used to build new protein from the protein of cells and tissue. Enzymes are protein and they control almost all biochemical reaction. Protein also act in immunity, transport, protein and growth. The slide continues the importance of protein. Protein help in muscles information and repair, and they maintain normal body functions. Some protein act as antibodies and product as viral infection. Some protein act as carrier and transport oxygen, hormones and nutrients in blood. This slide again reinforces that protein are essential in daily life. If protein intake the low for a long time, growth slow down and immunity become weak. The body may start using muscle protein to produce energy in regular deficiency. So regular protein intake is necessary for its health. Now we move to the disease related to protein malnutrition. Protein deficiency is common in children if the diet is not balanced. It causes weight loss, growth failure, weak immunity, and several in common cases. The main diseases are Kovacs and Maramamas. The slide explains the Kovacs symptoms and the treatment. In Kovacs, the shield loss of weight and growth stop, annual filled and belly swell due to edema. Immunity become weak, infection occur immediately. Check-in and care may be same color to that color. Kovacs can be treated by increased protein in diet along with balanced nutrient. Now we study marasmus. Marasmus usually occur in infant below 1 year of age. It happens due to deficiency of both calories and protein. The body becomes extremely thin and weak. The clay look like skin and bone due to several weight of class. This slide explains marasmus symptoms and treatment. Symptoms include several weight loss, growth failure, skin and hair color change, chronic diarrhea, and respiratory infection. Treatment is a balanced diet with protein, carbohydrate, fat, and essential nutrient. Treatment would be done carefully, gradually to help recovery. The slide mainly shows example for picture related to malnutrition. The key point is understand the difference. Coasio care often show bulimia or shivering. Will marasmus show severe wasting and a thin body. Both conditions need nutrient correction and medical care. Now we define malnutrition. Malnutrition means the body does not receive enough nutrient for proper function. It can be mild or severe and even life threatening in malnutrition and occur due to the starvation with low calories or due to deficiency of single nutrient like vitamin deficiency. This condition affects growth, immunity, and health. Malnutrition can also happen when the body cannot properly digest or absorb nutrients from food. This may occur in creating medical condition affecting digestion. Malnutrition remains a major global problem, especially in developing countries. Preventing malnutrition requires balanced diet, clean water, and food health support. Finally, we will learn about nutritional edema. Nutritional edema results from long time in protein loss and is common in farming area. Protein deficiency in adults is rare, but it can occur. Symptoms include weight loss reduced by body fat, frequent infection, lost stool, delayed food healing, and edema. A nutrient diet with soybean, milk, egg, and other protein-rich food and help prove protein deficiency in adult.